Let's take a look at red blood cells, or erythrocytes. Right? Erythro meaning red. This is showing you all the formed elements, right? So the red blood cells are right here. They're biconcave discs, meaning that, that's the shape of them, right? So it's concave right here, same on the other side. Here's a white blood cell, and then here's platelets. And platelets are actually pieces of a cell. But right now we're talking about erythrocytes, and erythrocytes really dominate the volume of blood. You have around 5 million red blood cells in a drop of blood, roughly. So, and then how many white blood cells? Thousands, right? So, really, red blood cells make up most of the formed elements. So, red blood cells are, I know this slide's a little bit confusing, and I crossed out some stuff so that you wouldn't try to memorize other things, but this is, so I'm looking at the part that I circled in red here, and red blood cells are kind of unique in that they don't have a nucleus. So they don't go through cell division like other cells do. They're born somewhere and they die somewhere, right? They're produced in the red bone marrow. So the bone marrow of your long bones, flat bones, like your your femur, your humerus, your sternum, and so that's where they're produced, and they're going to live for a certain amount of time, about 4 months, and then after that they are taken up by your liver and your spleen, and that's it, right? So here I've circled proerythroblast, and that's going to become a cell called a reticulocyte, and reticulocyte is where the nucleus, as you see, gets ejected, and then finally you end up with a red blood cell. So the reason I circled that up here is because, as you see, I wrote here, there is a hormone called erythropoietin, and erythropoietin ultimately increases red blood cells. And so that's something that's given in hospitals a lot. One of the formulations is called Epigen. And so when they talk about people doping, right, blood doping, that's what they're doing. They're taking Epigen, or they're taking erythropoietin. That's increasing the amount of red blood cells they have. So it's shooting up their hematocrit numbers, and and that means that they're going to increase their oxygen carrying capacity, which means that they can get oxygen around their body better, which makes them better cyclists or better whatever. So reticulocytes, sometimes people look at those in lab results, because we would expect so you've got like a total number of red blood cells, and then you would want reticulocytes to be like 1 to 2% of your total red blood cell count. Because one of the questions someone might have, if you have like a low hematocrit, is is the bone marrow producing more red blood cells? What's the problem? If your red blood cell amount is off, well, the problem could be coming from either end. Is the liver and spleen breaking them down like they should at the end of like 4 months, or are they being produced in sufficient numbers by the bone marrow, right? And so looking at reticulocytes in the blood could answer that question. But in any event, there you are with erythropoietin. So red blood cells are essentially masses of hemoglobin. And I wrote, this is wrong actually. I put here approximately 280 million is what I meant. About 280, 300 million hemoglobin molecules for each red blood cell. And each hemoglobin molecule can carry up to 4 molecules of oxygen. So if there's around 280 or 300 million molecules of hemoglobin on one red blood cell, and each one of those is carrying up to 4 molecules of oxygen, we've got like a billion molecules of oxygen being carried on one red blood cell. So it's pretty significant. So yeah, red blood cells are essentially masses of hemoglobin, and hemoglobin, as I wrote here, it's made from 2 parts. It's made from heme and globin. And as I wrote here, heme is a pigment and it has iron. So with heme, there's actually 2 parts. There's a pigment part and then there's the iron part. So it's like iron with pigment attached to it. And then globin is just a protein. So that's what hemoglobin is. And so what we're going to talk about in the next slide is how, what happens to red blood cells at the end of their life. And I'm going to go next to this slide, which looks like a lot of stuff written on it. So I made this slide to try to make it a little bit easier. So let's talk about the fate of red blood cells, something you definitely need to know. It's something that I often ask on tests. What is the fate of red blood cells? Or what are the fate of erythrocytes? Where are they made? Where are they broken down? Well, they're made in the red bone marrow. They're broken down in the liver and the spleen. If you notice here, look at the second bullet point, I've put the word phagocytize. So phagocytize, that means cell eating, right? Phagocytes are, the word PHAGO means to eat, and a cyte means a cell. So phagocytes are cells that eat things, right? And so we're using it here as a verb. So phagocytized in the liver means that in the liver and in the spleen, there's phagocytes that go and they eat the old, worn out red blood cells. And so let's go back to a red blood cell. What is it made of? Well, hemoglobin. What's hemoglobin made of? 3 parts. Pigment, iron, globin, right? The heme part is made from pigment and iron, and the globin, which is just protein. So the real question is when they get broken down, when red blood cells get broken down, what happened to these 3 parts? What happens to the globin? What happens to the iron? What happens to the heme, the pigment? And so the globin that's the easiest part to deal with, because your body recycles proteins all over. Your body is constantly recycling proteins. When you eat proteins, your body's taking those proteins, breaking them down, and building them back up to make other things. So proteins get broken down into amino acids and put them back together to make other proteins. So that's what happens to the globin. Globin's a protein, therefore, when we don't need it anymore in hemoglobin, we're going to break it down. We could use it to make more hemoglobin in the future, or we could maybe make something else. But the globin gets recycled. So now we're left with the heme part, and the heme is iron and pigment. So let's deal with the iron first. The iron also gets recycled. So I have here iron, and then I have like this arrow thing, and it's saying transferrin. So whenever you see F-E-R-R in a word, you think iron. So ferrin means, like the fer means iron, the trans means transport. So transferrin transports iron. It's a protein that's going to come, pick up that iron, and transport it. It's going to bring it over to a temporary storage site called ferritin, which is like another protein. So transferrin, the protein transferrin, picks up iron, takes it to a temporary storage protein called ferritin, and it's going to get held there. So you've got iron coming from your blood cells being broken down. But don't forget, you're eating food. So you've got iron coming from that steak that you ate or whatever you're eating, right? So that's coming in too. So you got iron coming in from like other sources, right? And you're storing it all in the temporary storage site, ferritin. Transferrin, after you get a bunch of of iron stored up, transferrin is going to come back up, pick up all that iron, and bring it over to the red bone marrow. Because the red bone marrow is where you make red blood cells. So we're going to take that iron back to the red bone marrow so we can make more red blood cells. So now we're left with the pigment, and that's the most difficult part. Pigment is not recycled. So the pigment gets it gets broken down into other pigments until it eventually gets excreted, right? So you have a pigment called biliverdin, which I didn't put up here. That's going to become bilirubin, and bilirubin is a common lab test because looking at bilirubin levels can give you some idea about how fast or how slow your blood cells are getting broken up. So that's kind of the other end of it, right? We could look at the liver and we could look at bilirubin and we could see, what's going on with red blood cells. Or, it's also indicative of liver function. But anyway, bilirubin turns into another pigment called urobilinogen. So now from the liver, it's going to eventually end up in something called, it's going to end up in your bile, which is made by the liver and it ends up going into your small intestines. That part, we can get to that later, but now it ends up in your digestive tract. That's what I'm trying to say. And so some of that urobilinogen is going to get turned into another pigment, brownish pigment called stercobilin. That's going to continue down your digestive tract. That is what is coloring your feces brown. Some of that pigment is going to get turned into urobilin at the kidneys, and that's a yellow pigment, and that's going to color your urine. So the color of your urine and your feces was the color of your from your red blood cells, right? So here's another version of what we just talked about, and you could kind of follow it along, and it's the same thing, right? So we take the globin first. It's up here where I'm pointing now. Globin gets broken down into amino acids and reused again. So that's proteins getting recycled. So done, right? And then here is the iron part of it. So you see, so this is the heme, and you see two arrows coming out of the heme. Here's one arrow going this way, and then here's one arrow going this way. So this is the iron part, and that's the pigment part, right? So iron, transferrin picks it up, stores it as ferritin. Transferrin is going to pick it up again and take it to the red bone marrow. And then when you follow the pigments, biliverdin, bilirubin, and these are kind of giving you the colors of the pigment as well, right? So there's bilirubin in your liver. It's going to go into bile, and it's going to end up in your small intestine. And then you follow it through. There's urobilinogen, and then urobilinogen splits into urobilin and stercobilin. That's a pain, I know, because it's a bunch of new words that you have to learn and memorize. But yeah, there's no easy way to do it. You just got to do it, right? Oh, and see, I wrote up here, liver test of bilirubin. Elevated may be hemolysis, right? So let's look at these words. There's lysis. Lysis means to break down something. Hemo is talking about your blood. So your blood cells are breaking down. If it's too high bilirubin, that means blood cells are getting broken up too quickly. So anyway, there you go. That's red blood cells. I think in another, because we're at about 15 minutes now, so I think another, I'll put up another video about white blood cells. I'll probably put white blood cells and platelets together. So I'm going to stop this video here.